Hey everyone, I'm Dr. Steve Carroll, and I want to welcome you to Sensation and Perception. What do you see? Well, isn't part of your brain telling you that that's a face? I mean, it's not a face, it's rock. Well, it's not even rock. These are pixels that are lighting up on your screen. But somehow you've arranged them to tell you that this is a rock that is trying to look like a face. What's that all about? Why is your brain lying to you? I mean, it's not a face, right? I mean, clearly it's not a face. Why is your brain lying to you? What do you see? Why is your brain lying to you? What do you see? It's Slender Man. It's a tree version of Slender Man. No, it's not. It's just a tree. I want you to look at this. I want you to pay particular attention to the two squares labeled A and B. Now, this is a trick. Okay, I'm telling you this is a trick. I've seen this 1,000 times, and I'm telling you it's a trick. But I want you to be completely honest with yourself. Which square looks darker? I mean, A obviously looks darker. If A doesn't look darker to you, then there's something strange going on with your visual system. But let's see what's happening. I've taken away everything except for those two squares. And now we're zooming in. I haven't touched the picture. I haven't altered the picture. All I did was zoom in on it. Now let's take away the rest of that pattern. Which square looks darker? They look identical, don't they? Because they are identical. In fact, they are exactly the same color. So now that you know that this is true, now that you know that this is a trick, tell your brain A and B are the same color. Okay, you know this. I'm letting you in on the secret. A and B are the same color. You can't see it that way, can you? Your brain is lying to you. Look at the two center circles. Doesn't the one on the left look bigger than the one on the right? I mean, you've probably seen this illusion before. You know that they're exactly the same size. But it still looks bigger. Why is your brain lying to you? Okay, let's go over the syllabus. You should download it from Brightspace right now. You can see when all of your assignments are due. You have an assignment that's actually due every lecture day. Okay, so if you're watching this on September 15th, you actually have an assignment due at 5 p.m. tonight. This is how you get a hold of me. So stephen.carroll@smu.ca. So here's the thing. I'm actually teaching a lot of courses remotely this term, which is a little more challenging than teaching all those courses in person. Believe it or not, it's actually harder to mark things online than it is to mark them with just a red pen. So I'm teaching Intro Stats, the Intro Stats Lab, Advanced Stats, the Advanced Stats Lab, and this course. So I pretty much am marking my whole life from now until December 31st. But I'm going to set aside Tuesdays. So Tuesdays is when I'm going to deal with you guys. Okay, so Tuesdays I'm setting aside to answer your emails. So when you email me, when you try to get a hold of me, if, of course, I'm not a robot, if I have time, I'll email you earlier. But generally speaking, Tuesdays is when I'm going to focus on sensation and perception emails. Here's your textbook. It's actually pretty good. Now, when I actually taught this course as a special topics, I called it Sensation, Perception, and Psychophysics. I didn't have a textbook. I tried to get as many free online readings as possible. And so what I got the class to do were read a bunch of articles. The problem is that course was being offered as a fourth year, And so all of the students in the class had already taken all of their stats courses. And so they understood how to read scientific articles. The problem is, this is a second year course, and I can't be sure that you've taken any stats courses. So I can't give you something as advanced as a scientific article as you're reading. So unfortunately, that means I had to choose a textbook. And I know textbooks are expensive, so I'm sorry about that. But that said, this is a pretty good one. I've actually enjoyed reading this textbook. So there it is. Get an online version if you can. You're going to be evaluated through some weekly assignments and a final exam. So every week, you're going to owe me a written assignment, and those assignments all together are going to be worth 70% of your final grade. And then we're going to have a final exam, which will be worth 30% of the grade. I do not know what the format of that final exam is going to be yet. Exams are actually pretty challenging for a remote course, so I'm still working that out. But all of your assignments should be available online by the time this course starts September 15th. I'd like you to submit your assignments as either Word documents or PDFs. Okay? Please use the following naming convention. So your last name, dash, first name, dash, then say PSYC 2110, dash, and then ass, whatever the assignment is. So there, you have my permission to write ass. I want you to follow this naming convention because, as I mentioned, I'm going to be getting a lot of assignments from a lot of different people in a lot of different classes. And if you don't want yours to get lost, yeah, please follow the naming convention so I know where to file this thing. A late penalty of 10% per day will be assigned to late assignments. Assignments that are handed in more than 3 days late will receive a grade of 0. I'm sorry I have to be mean that way, but again, I've got so many assignments coming in, I don't really have time to wait. Okay, so let's talk about assignment 1. Nothing too difficult. I'm going to get you to build a creature. I'm actually going to get you to design the nervous system of a creature. It's going to be a simple creature. I mean, it's your first day as acting as God here. So let's make it simple. Simple in that it's going to need to follow some very simple rules in order to survive. So this is going to be like a plant. So the organism is going to use sunlight to create the energy it needs to power itself. It's going to need to be able to move, so it's going to need to be able to follow sun throughout the day, and it's going to need to avoid sneaky predators who are going to shine phosphorescent lights to try to lure the creature towards their mouths. So think about this as a little, just a simple organism that lives in a pond of water that is going to swim around to follow the sun so that it can feed itself, and at the same time, avoid predators. And I'm going to use this thing as a model. So I actually found this particular fossil, and it's absolutely fascinating to me. So look at the top right of the screen. You'll see this oval shape, and it's got 2 flagella that seem to be coming out from its bottom. Okay, so this is going to be our base. And look, it's even got this little nucleus thing right in the middle of the oval. So this is going to be our model, our template. So here we go. So here's our basic model. So this is the structure of our organism. Now, what does it need? So it needs to be able to accomplish those tasks that I talked about. So what's it going to need? Well, obviously, it's going to need to have some cells, some sort of receptors that are sensitive to light, right? If it's going to follow the light, it somehow is going to have to be able to sense the light. Is that biologically plausible? Well, yeah, I'm not going to get you to just make something up. This isn't science fiction. The processes can be quite complicated, but there are many biological systems that respond to sunlight, and that actually includes you. There are all kinds of systems in your body that respond to sunlight. There are chemical reactions in your body. Sunlight causes, or at least sparks, these chemical reactions. Now, we're going to get to that in greater detail when we talk about vision, but yes, I just want to point out that this is biologically possible. Hell, plants, right? Plants all respond to sunlight. I mean, that's what they do. So we're going to have this cluster of cells in the middle that are going to respond to sunlight. And those cells are going to send signals out to the flagella. So somehow that little cluster of dots in the middle is going to respond to sunlight. And the way it's going to respond to sunlight is by telling those little flagella to move. They're going to wiggle their legs so it can move around. But how would that work? Suppose the cells can, in some way, respond when they come into contact with light. And by light, I mean electromagnetic radiation. And we'll talk about that next week. But how can they use that reaction to move the flagella? This is what sensation is. So when I talk about sensation, what I mean is the nervous system's initial response to some kind of external stimulus. Okay? So when something happens in your environment that causes a nervous system reaction, that's sensation. It's your nervous system's initial response to contact from something in the environment. And I mean contact literally. It's about touch. The whole thing is about touch. We think about, sight as our primary sense, or hearing is very important. But these are all responding in the same way to touch. So photons touch the photoreceptors at the back of your eyes, and that sparks sight. Air pushes against the tympanic membrane in your ear canal. So molecules in the air are touching the tympanic membrane, and that starts hearing. For taste and smell, molecules come into contact with receptor sites in your mouth and your nose. So these molecules touch parts of your nervous system in your mouth and in your nose, and that starts those processes. And touch, I mean, this one's more literal, mechanoreceptors respond to things distorting the shape of your skin. So when something bends your skin, you feel it. That's what touch is. Well, sort of. Touch is weird. We'll get to touch later. Here's something I want you to think about throughout this course. All right, a big chunk of this is going to be biology. And biology depends, or at least is largely explained, 99.999% of it is explained by evolution. All right? All of our sensory systems respond to physical contact with the outside world. And as such, they're all actually quite similar to one another in many ways. It's almost as if our species began with a single sensory system that bifurcated, that sort of became different sensory systems over the millions of years of our evolution. That we started with one way to perceive the world, or sense the world, not perceive, sense the world, and that it specialized into different kinds of ways to sense the world. Evolution explains a hell of a lot. Okay, but how? How can touch start awareness? Well, here's the thing. For all the beautiful complexity of the human mind, it is made up of some really tiny, stupid little things called neurons. Here's a picture of a neuron. They're just cells. They're cells that have some rather unique features, but they're just cells. So tiny, tiny single cells. And essentially the way they work is if they come into contact with certain chemicals, they get excited and they splooch out a bunch of chemicals of their own. That's it. So they just sort of reach out into space, and if they come into contact with chemicals, they send out a chemical signal of their own. That's it. That's you. That's your whole brain. Here's a simplified two-dimensional version of what's going on. So these are the things that we're going to care about here. And I want to spend a little bit of time just talking about that area right there called the synapse. Let me just give you a brief idea of what happens here. Here's the soma of the neuron. It just contains the organelles, all the things that the neuron needs to keep itself alive. Okay, all the little fiddly biological chemical things that keep it functioning. And it has these things called dendrites, these little branches, right? Dendrite from tree. These little branches that reach off into space. Now what happens is there are these little chemicals that are just floating around in space. Well, not really. I'll explain in a sec. When these chemicals come into contact with the dendrite, it gets the neuron a little excited. And if enough chemicals come into contact with the dendrite, it gets more and more excited, and eventually, boom, it sends off its own signal. So if it gets excited, it sends off its own signal. So it will splurch out a bunch of chemicals of its own into this area called the synapse. Now, as you can see, the synapse connects, well, sort of, one neuron to another neuron. They're not actually physically touching, but they're in very close proximity. So it's going to send out its own chemical signals that in turn might activate this neuron. And that's how they work. They're just little light switches. When this light switch turns on, there's a chance it's going to make this light switch turn on. And when this light switch is off, it can't make this one turn on. That's it. Your brain is made up of light switches. But let's start with the synapse. So here's what's going on. So we have a presynaptic neuron and a postsynaptic neuron. So this up here is the presynaptic neuron. Okay, by presynapse, I mean it is the one that comes before in the synapse. And the synapse is just this little space right here. A synapse is just a space between neurons. So we have a presynaptic neuron and a postsynaptic neuron. So down here, this is the dendrite of a postsynaptic neuron. And this is what's called the axon of the presynaptic neuron. So the axon is where the chemical signal gets splurched out of. Okay. You see these little things? These little bubbles? These little bubbles contain chemicals. Okay. So these little chemicals are in these bubbles. The bubbles come into contact with the wall of the terminal button, here, the axon's terminal button. It fuses, so the bubble will fuse, and in fusing it will release its chemicals into the synapse. The chemicals will float around in the synapse until they come into contact with what are called receptor sites. Okay, that's the gist. So synaptic vesicles are packages that contain the neurotransmitter molecules, so the chemicals, the chemical signals, and they fuse to the cell of the membrane. They fuse to the cell membrane, and then they dispense their neurotransmitters through a process called exocytosis. Okay, so I've given you a list of videos for each of these videos that I'm making. I'm also going to give you a list of YouTube videos that I want you to go and watch as part of the lecture. If we were doing this live, I would just show them in class, but I think you should go and, when I post these, you should go and look at them because they're going to help you understand exactly what's going on. The neurotransmitter molecules are a chemical substance that is released from the presynaptic axon terminal and they act on the postsynaptic cell. Okay, so the neurotransmitter molecules are just these little chemicals that might cause the postsynaptic neuron to fire. The axon terminal, the terminal button, is that big bulby shape, right? This big bulb shape right here, that's the axon terminal. It forms the end of an axon that forms a synapse with the postsynaptic dendrite. Okay, that bit right there that I've highlighted, that's the synapse. The synapse is also known as the synaptic gap or the synaptic cleft. It's just a small space. In fact, the synapse is nothing. It's a blank area. It's a space where the neurotransmitters can float. It's the name we give to the fluid-filled gap. It's not sci-fi techno magic. So you get all these books and movies where they talk about the synapses this and synapse that, and oh, the aliens are affecting the synapse of the robot and blah, blah, blah. The synapse is a hole. There's nothing there. Anyway, just thought I'd rant. A receptor. So when the neurotransmitters are just sort of floating through space, eventually they can come into contact with a special molecule on the postsynaptic neuron. It's called the binding site. Now, certain chemicals, so certain neurotransmitters, bind to certain receptor sites. So different chemicals can cause the postsynaptic neuron to act in different ways because they bond with different molecules on the postsynaptic neuron. Now, when a neuron gets excited, it releases chemicals into the synaptic cleft. Those chemicals float around until they attach themselves to specific receptor sites. So it's like a lock and key. Only certain chemicals will fit to certain specific receptor sites. So in the case of our organism, remember you're building an organism? In the case of the organism that you're going to build, one type of cell will unlock only in the presence of light radiation. So we're going to use photons to cause this chemical reaction. The idea is when a neurotransmitter locks into the receptor site, it opens up an ion channel. So what actually happens is when the chemical reaches the binding site, so the molecule, it distorts that molecule. It causes that molecule to bend. And in bending, it actually opens up a channel. Okay? Now, when it opens up that particular channel, it allows other chemicals to rush into the space. So an ion is a molecule that has either a positive or a negative electrical charge. So if an ion has a positive charge, it's called a cation. If it has a negative charge, it's called an anion. Positively charged cations have more protons than electrons. So this is what we're talking about. So we're talking about atoms and molecules. Okay, so they're made up of neutrons, which have neutral charges, protons, which have positive charges, and electrons, which have negative charges. So an ion is a cation if it has more protons than electrons, meaning it has an overall positive charge. If it has more electrons than protons, then it has an overall negative charge, and it's an anion. The flow of ions cause either excitatory postsynaptic potentials, so EPSPs, also called depolarizations, or inhibitory postsynaptic potentials, so IPSPs, or what we call hyperpolarizations in the receiving cell. Okay. So think about it this way. It can either excite or inhibit. So some of the chemicals, when they float down through the synaptic cleft and they bind to the receptor sites, encourage that other neuron to get excited and to fire its own signal. But other times, the chemicals will float down through the synaptic cleft and they'll bind to the postsynaptic receptor site, and they will instead discourage, they will inhibit that postsynaptic neuron from firing. So some neurons encourage other neurons to fire, but some neurons encourage other neurons to not fire. So in other words, some neurotransmitters, so some of the chemicals that are inside the neurons that are dispersed from the neurons, are more cause other neurons to become more active. So for example, glutamate. Glutamate is one of these. And by more active, I mean they're causing the postsynaptic neuron to be more likely to fire off their own chemical signals. But other neurotransmitters, other chemicals like GABA, encourage other neurons to become less active. Neurotransmitters do this by controlling which ions, so either cations or anions, they're controlling which ones enter the cell and which ones are forced out of the cell. So for example, glutamate causes an excitatory postsynaptic potential. So when it binds to the receptor site, it opens up this channel and causes sodium to enter the neuron and calcium to enter the neuron. So the result is that glutamate opens up this channel that lets all of these positive ions enter the cell. So that as a result, because all of these positively charged ions have rushed into the postsynaptic neuron, the result is that that neuron now has a positive charge. GABA, for instance, when it binds to a receptor site, it causes potassium, a positively charged ion, to leave the postsynaptic neuron. And it also causes chloride, a negatively charged ion, to enter. So the postsynaptic neuron has lost some positive ions and has gained some negative ions. So the overall result is that the cell becomes more negative. So what's all this positive versus negative stuff? Well, you might have heard the expression that opposites attract. That actually comes from physics, where positively charged ions are drawn towards negatively charged ions, and vice versa. But positively charged ions repel other positively charged ions, and negatively charged ions repel other negatively charged ions. Well, so what? Life depends on movement. So if there was no movement, the universe would not contain life. There are lots of natural forces that cause things to move, but we're only going to care about two: diffusion and electrostatic pressure. Okay? Now, this natural inclination for positively charged ions to move towards negatively charged ions, and vice versa, and for positively charged ions to be pushed away from other positively charged ions. So we have this natural source of movement that actually causes your nervous system to work. Okay, so let's talk about communication within and between the neurons. Okay, so here we have a little diagram where we have intracellular fluid and extracellular fluid, and we've got some little ions floating in there. Now you can see some are positive and some are negative. So potassium is a positively charged ion found in abundance inside the cell. Okay, so the cell has this positively charged ion within it. Chloride is a negatively charged ion that's found outside the cell. Sodium is a positively charged ion found in abundance outside the cell. So now the interaction between potassium and chloride and sodium is, in large part, how your neurons are able to communicate with one another. Your brain works because of potassium and sodium chloride, which is why I cannot recommend chips highly enough. Think about it, right? Potatoes, potassium, and salt, sodium chloride. That's it. Chips are brain food. Eat all the chips. Okay. Diffusion is the tendency for molecules to move from areas of high concentration to areas of low concentration. So here's another thing. If you have a lot of molecules all packed together in a tight area, and you suddenly open up a new space for them to go, they will move out into that space. They will spread out. They will try to maximize the distance between themselves. There are more potassium ions within the cell than outside the cell. There are more chloride and sodium ions outside the cell than inside the cell. So because of diffusion, potassium wants to push out and chloride and sodium want to push in. Okay, so regardless of the positive attracted to negative, what's happening is that there's just so much in one area that it wants to move into a new area. Electrostatic pressure is the force by which two ions with similar charges will repel each other, and the force by which two ions with opposite charges will be attracted to one another. So sodium and potassium will repel one another, right? Because they're both positively charged. So potassium wants to get away from sodium. Sodium wants to get away from potassium. Chloride and the other anions within the cell repel each other. So anything that's negatively charged wants to push away from anything else that's negatively charged. On the other hand, the sodium and the potassium are attracted to the chloride and the other anions, right? That's why we have sodium chloride, table salt, sodium chloride, because the sodium and the chloride is drawn to one another. Now, our presynaptic neuron has released a molecule into the synaptic cleft, and it has binded to a receptor site. And that, in turn, has distorted that receptor site molecule, and it has created a hole. So now there's this little hole. Well, that's called an ion gate, and that ion gate has opened. And now we've got all of this tension going on. The positives hate the positives, and they love the negatives, and there's all this romance and stuff. So that gate has opened up, and now all kinds of action can take place. Positive is pulled towards negative. Positive is pushed away from positive. Negative is pushed away from negative. When all of the ion channels are closed, so when that gate is shut, the cell has what we call a negative resting potential. Okay, what am I talking about? Well, it has a negative resting potential. The cell tends towards a negative state. Okay, it tends to be more negatively charged than positively charged. The reason is because potassium can actually pass through the cell membrane pretty easily, even without the channels. So even when that gate's closed, potassium has found a sneaky way to get out of the cell. Chloride and sodium can also pass through the membrane, but with more difficulty. They can't quite get through the secret gate. The other anions, big old negative anions, they can't pass through the cell even when the channels are open. So even when the channels are closed, there is still some movement of these ions. But in general, the anions can't leave the cell. So that means that with everything else being able to move around, the cell, in general, has a negative resting potential because of all those other anions that can't leave no matter what. The net result is that the cell holds a negative charge, meaning there are more negative ions inside than outside. And we can measure how much. It has a resting potential of negative 70 millivolts. Incidentally, a millivolt is 1,000th of a volt. So what's a volt? It's a measure of force. If you ever play around with electronics, which I highly recommend you do because it's a lot of fun, a volt is just a measure of force, how much work can be done. So with all of this pressure, any neurotransmitter molecule that forces open one of the ion channels causes a sudden rush of movement, and the molecules all start to exchange places. And movement equals life. Okay, so here's the deal. So you can see from this little graph that the action, so we're calling this the action potential, so the potential for action. At the beginning, so on the left side of this chart, you see that the resting membrane potential is negative, negative 70 millivolts. The action potential is dependent upon the exchange of ions that lie on either side of the membrane. So excitatory neurotransmitters open the channels and cause the membrane to shift from 70 millivolts to about negative 50 millivolts. It's still negative on the whole, but there is a shift. Clearly there's some movement going on. So in other words, the intracellular fluid within the neuron becomes less negative and more positive. Still negative on the whole, but generally less negative. When that happens, so when it gets to about this negative 50 millivolts, the sodium channels open. So when the cell reaches this threshold of excitation, the sodium channels pop open. And when that happens, things really start to move. Remember, sodium is a positively charged ion. So if the sodium can suddenly rush into the cell is suddenly going to have a positive charge. It's going to depolarize. Both diffusion and electrostatic pressure push the sodium into the cell. So when the sodium channels open, sodium is under lots of pressure to rush across the membrane and into the cell. Sodium moves very quickly across the membrane and into the ion channels, like very quickly. That causes the potassium channels to open. As the charge on the inside of the cell is increasing due to the influx of sodium, the potassium channels open and begin to let the potassium back out of the cell. At the peak of the action potential, the sodium channels begin to close. Okay, so now the cell has become very positively charged. The sodium channels begin to close. Diffusion and electrostatic pressure start forcing the potassium out. So you have all this potassium has rushed into the cell, but now potassium doesn't like potassium, so it tries to spread out, so it starts to push back out. And because of all of that potassium and sodium, it has forced the cell to become positively charged, and so the positively charged ions are no longer happy inside the cell, they want to leave. At that point, the cell undergoes a brief hyperpolarization. So that means it rests, so once it's fired, it has to take a little nap. So it's done its thing, and now it has to take a nap. It can't fire again for a brief period. Okay? All right. This gist, okay, we'll get to that in a second. This simply means that once an action potential begins, it will not diminish or stop in any way. Okay, so it's an all or nothing thing. Once the neuron fires, it will fire. It can't stop until it is finished. It's an all or nothing thing. The action potential starts at one small part of the axon and then propagates down the shaft towards the terminal button. So remember, the receiving end, so the receiving neuron, this is actually happening at its dendrite. The action potential, well, and then eventually gets down to the axon of the receiving neuron. So the action potential occurs at the top of the axon, so that long branch that ends in the terminal button. The action potential, it occurs at the top of the axon, and that exchange of ions causes channels to open in an adjacent part of the axon, which causes another action potential, which in turn, et cetera, et cetera. So let's see if I have a graph here. Yeah, we know a lot of this because of Hodgkin and Huxley's studies into the giant squid. A giant axon in a giant squid. That's actually the name of the axon. It's not just, wow, what a giant axon. No, it's actually called the giant axon. And they studied this, so because it was so big, they could actually look at it. But the idea is this. So once this depolarization event happens in one part of the axon, so you get this rush of positive ions into the cell, that causes other gates that are nearby to modify their behaviors. So the rush of sodium into one part of the axon, the top of the axon, will cause an adjacent part of the axon to also have that response. So when the sodium gate opens on the left side of the axon, something just to the right of that will also open up. And that's how the signal gets propagated down the length of the axon. So one action potential causes an action potential in an adjacent part of the axon, then that will cause an action potential in an adjacent part of the axon, etc. And so the chemical signal gets propagated all the way down the axon until it reaches its own terminal button. Once the action potential reaches the end of the terminal button, neurotransmitter gets released into the synapse and it passes the message on to the next cell. So now this action potential is zipped all the way down the axon, and as a result, what happens to the terminal button is when it gets excited, it releases all of these chemicals into the synaptic cleft. Now that those chemicals are released, there's a chance that they're going to cause the same thing to happen to another neuron. These chemicals, though, can create either excitatory postsynaptic potentials or inhibitory postsynaptic potentials. So they can either encourage the neuron to fire by opening up the gates or inhibiting them by blocking the gates. So what some of these chemicals do is, rather than opening up the gate, they block the keyhole. So another neuron can't actually open the gate. And the thing to keep in mind is, this rarely happens singularly. This isn't about one neuron talking to another single neuron. It's bunches of neurons communicating with bunches of neurons. So for one particular neuron, it's actually receiving signals from hundreds, perhaps, of the neurons that surround it. Some of them are excitatory, some of them are inhibitory. So there's this calculus that's going on. Some of the neurons don't want the neuron to fire. Some of the neurons want it to fire. So there's this fight that goes on. Most often a cell is receiving multiple messages. So EPSPs and IPSPs have to be added up. There is an inherent calculus within your nervous system. Even if you don't like math, your neurons are always doing math. They're adding things up. Am I more positive or more negative? Now, if those chemicals that are in the synaptic cleft are just left to float there, it's going to cause a problem. Once neurotransmitter is released into the synapse, it's going to continue to bind to the receptor sites and cause that neuron to fire over and over and over and over and over again. Well, you don't want that. It can be dangerous. It can lead to overexcitation. So the brain actually has some different ways to remove those neurotransmitters. So one thing it does is it has enzymes. So enzymes are proteins that cause chemical reactions. So there are these enzymes that just kind of float around in the synapse. They find neurotransmitters and they break them down before they have a chance to cause the postsynaptic neuron to fire again. Another thing is called reuptake. So sometimes those little chemicals, those neurotransmitters that are floating around in the cleft, will float back up towards the presynaptic neuron. And when they do that, they sometimes get reabsorbed. So we call that reuptake. And when they're reabsorbed, they can be recycled, they can be repackaged, and used again. So let's look at the rest of the neuron now. So dendrites. This is where it all starts. Dendrites are the input. So it's the part of the neuron that is sensitive to the neuron's environment. It's the main input of the neuron. The soma is the body of the neuron, the cell body. It contains the nucleus, and it directs protein synthesis for the cell, all the little functions that keep the neuron alive. And that nucleus, it's kind of like the brain of the neuron. Well, no, because it's too stupid to be a brain. But it contains genetic material for the cell. The axon is the part of the neutron that extends from the soma. So that's the main output from the neuron. So the dendrites receive the chemical signals. Right? That's where the action potential starts. When it gets excited enough, it'll shoot a signal, an action potential down the axon. And that becomes the main output. The myelin sheath, I'll show you a picture in a second, is a lipid, like fat, lipid-based substance around the axon of a neuron that serves as an insulator. Right? So you know how you have anything, any wire that you have, when you plug your phone in at night, that wire is covered in a plastic sheath or a rubber sheath. That's because that is an insulator. So the electrons can't jump out of the wire onto something else because it doesn't conduct electrons very well. This myelin sheath is going to have little gaps in it. And those gaps are called the nodes of Ranvier. And they're actually very important. So what happens is, what's going on with my slides here? Yeah, that's weird. Let's get them all. Okay. Myelin speeds the process. So the action potentials are only happening at each node of Ranvier. So because of this lipid insulation, it forces all the chemicals to zip down the axon until they reach another uninsulated part, and that's where the action potential occurs. So the action potential doesn't actually have to happen at every little micromillimeter all along the axon. These nodes of Ranvier say, well, we'll speed up the process. If an action potential occurs at one link, we can just zip down to the next empty space and we'll have an action potential there. So rather than, for example, in this diagram having thousands of little gate opening events, you would only have the gate opening events at each of the nodes. A breakdown in that sheath is actually, it inhibits efficient communication between the cells. That's actually what multiple sclerosis is. Multiple sclerosis is about a breakdown of the myelin sheath. So what's happening is that you don't have this efficient communication between the neurons anymore. When that sheath goes away, communication between the neurons becomes very slow and clunky. Okay, so there are all the parts of the neuron. You should know what each of them are, what they all do. That's perfect test material. And this as well. Okay, so you should be able to identify everything there. Now, there are different kinds of neurons in the brain. So we have sensory neurons receive information from the outside world. So instead of responding to neurotransmitters, they respond to other forms of stimuli. So the idea is the same, except that the receptor sites don't respond to a chemical, they respond to something else. So in the case of our organism, we're going to have one that responds to photons. That's the sensation part of the class. Okay. Interneurons process the sensory input from our environment into meaningful representations. They help plan, well, they don't, well, it's complicated, but they plan the appropriate behavioral response, and they connect to motor neurons to execute these behavioral plans. So in the case of a human with our complicated brains, these interneurons take all of this sensory information and then they turn it into meaning. That's the perception part of the class. So sensation is when your nervous system responds to the environment. Perception is when you ascribe meaning to those sensations. Motor neurons initiate movement and behavior. And that's someone else's class. This isn't sensation, perception, and running around the block. That's someone else's problem. Bipolar neurons are involved in sensory perception, such as the perception of light in the retina of the eye. We'll talk about that next week. They have only one axon and one dendrite. So in some ways, they're a lot simpler. You have one input, one output, as opposed to some neurons that have many, many inputs and only one output. These are useful. They're useful for quickly acquiring and passing sensory information to different parts of the brain. Unipolar neurons are kind of interesting. They're only used, well, they're useful for just sending information forward. So they're just passing the message along. They only have one axon and they have no dendrites. So it's kind of interesting. They're involved in transmission of physiological information from the body's periphery up through the spinal cord to the brain. So you have a central nervous system, which is your brain and your spinal cord. And you have a peripheral nervous system, which is the other nerves, and they send information to the spine. And that's where you find a lot of unipolar neurons. Multipolar neurons are the kinds that we've just talked about. So they're more common, the most common, and they have lots of dendrites and only one axon. Okay, that's enough biology for today. I had to go through that because it's a second-year course, and I can't know for certain that you've actually taken a biology class before. But this whole thing is about neurons, so I had to take some time to explain neurons. Okay, well, why don't we take a break before we go to the next video? Go grab yourself some caffeine, go walk around the block, get some chips, because you know chips, right? Brain food. And I'll see you in the next part 2 of today's lecture.